Okay, hello, how are you everyone? Nobody's here. Uh, Today, I think I have to keep reminding myself that I have to stay closer to this area microphone because every time I screw up at least one thing. Today I forgot to bring my this microphone thingy. So if I move away from this computer source, probably my voice will just drop in the recordings, not for your actual live lecture. It's no problem. So, okay, today we are moving into new chapter, which is about the cell structure. Actually, the main objective of this chapter is to look at and understand the various function, structural function of the eukaryotic cellular organelles. But to do so, we have a little bit of the introductory background information, which we'll take a look at first, the prokaryotic things, then move on to eukaryote. So that what's the actual difference, so that we can appreciate more about the eukaryotic complexity present in the organelles. So, having said that, let's move on. So in order to take a look at the cell seriously, then what we need is a actual device, tool. How are we going to take a look at inside of the cell precisely in more detail? That comes to the microscope. Okay. So before the microscope, first let me introduce first this famous cell theory, which personally I believe that what this is really something great, big deal, because okay, this cell theory was formulated circa about because these two people credited to this famous so-called cell theory two German gentlemen and scientists lived this period or something 19 mid 19th century. So probably in the middle of the 19th centuries, this cell theory was introduced and recognized. So it goes like this: three principles of. It reads like cells are the fundamental units of life. Duh, of course, everybody knows that. But back then, probably some people, at least to some people, it was quite new thing. Oh, really? But nowadays, everybody agrees on this. Of course, cells are the fundamental unit of life. That's why in the very first lecture, I just stated that cell is the basic unit of life, didn't I? Yeah. And a little more important than the first doctrine, so to speak, is all life forms are made of cells. Yeah, nothing else. What about viruses? That's why we consider viruses are non-living, which not that important or significant, and that matters. But anyway, that's how we deal with them. And actually, this is the key thing. It reminds you of something like the conflict between the biogenesis versus spontaneous generation theory. Also, you can see in this the final statement, which goes, all cells are derived from existing cells by cell division. Yes, which is of course so obvious these days. What else? But back then, probably that must have been something very quite revolutionary or new. And since then on, this whole cell theory has been established and based upon it. Everyone think about around which, at least as far as this cell function and structure goes on. But the main thing that I introduced this was the reason is because were they able to, in particular, around the mid-19th century, they finally were able to come up with this so-called cell theory. Yeah, let's just consider that, of course, that's something very important. Well, let's clap it even. Okay, good job. But why and how only then? It was merely the result of the advancement of technological advancement of this microscope. Without it, without being able to look at, actually, examine under any device called microscope, how am I going to be able to state stuff like that? Actually, when those days, when the first real functional usable microscope was introduced to the European society, those people, important people, as so-called the high society, were quite shocked to discover that, like, sample taken from small pond, okay, there was so many moving stuff, crazy like moving things, in seemingly really very pure, quite calm sample of water taken from pond. So that much was, at least that much was a cultural shock. Okay, there are so many different forms of life, so to speak, microorganisms, bacteria, and some other eukaryotic microorganisms were only visible under the microscope. And then they start thinking about, okay, what is this going on? How many different kind of life forms are there out there in nature? And how do we actually go with it, define those? And then finally came to this conclusion, okay, everybody, including us and all other multicellular organisms and those tiny little bit of single-celled organisms that we just recently discovered under the microscope are all basically had common thing, which was cellular basis. All life forms were made basically by cell, the unit of life. So microscope was that much like instrumental, I should say. So the first form of microscope, mostly commonly people consider this Robert Hooke, the British scientist, was the first line of a microscope. Actually, it was not the first microscope, but anyhow, this type of cell-like structure, it was actually that cell, cork, the tree's surface. But after discovering this under the microscope, Robert Hooke named this as a cell. So he was the first one who called this cell as a cell. Because this structure was very similar to those compartments in the monarchy in the Westminster. Actual practical matter, because I have something in mind. My agenda is finally later, at the end of this talk of the microscope, probably, hopefully, my true agenda, hidden agenda about this topic of microscope can be naturally revealed to you. But let me just start with this. How can you see, actually, practically speaking, some little things? For example, bacteria. Anybody who took the college level or even the advanced high school level microbiology laboratory exercise, you would already know. Have you ever tried to see any small bacteria under the microscope in your laboratory exercise? Were you able to see readily? No? Why not? Cells were there, so you were supposed to be able to see bacteria under the microscope. Well, then you are not able to actually. Why? Exactly this. They're not simply staying stuck, stand waiting to be observed. They are living things. They keep moving, dynamic. So how can we tell them, okay, don't move, stay there, I'm going to look at you? How can we deliver our intentional message? So we have to fix them. Fix, and then we have to increase their contrast, the image. Because the living things, even microscope is basically what? The magnifying glass. Using the lens, you are simply magnifying the images so that you may be able to now enlarge the object so that now you may be able to see. However, you cannot increase the individual contrast, so still they may be very blurry, so you may not be able to actually distinguish which ones are which. Are these there's a cell boundary or some type of a water droplet? How do you know? So you have to introduce some kind of like distinguishing in terms of staining. And also you have the staining, coloring the sample, living sample, actually do these two jobs for us at the same time. By staining this living microorganism to visualize, in order to visualize under the microscope, then instantly, usually those are staining, those are chemicals. They will kill. They will kill those microorganisms, the living cells, and then also those chemicals will be able to fix. So that stabilize them, don't move, they were just hung in there where they were, is basically the fixing procedure. So staining will be able to, practically speaking, do both. So now we can see them. But so most of you obviously then can think of some negative effect of this staining to see the sample under the microscope, which is now you are looking at already dead sample. And also by staining them, coloring, which is artificially developed chemicals, although with different colors of the staining solution, you may be able to see much better to distinguish different parts, but at the same time, it is an artifact. It is not natural thing. It may actually, what you see under the microscope after the stain may not actually represent the real, the image in life. There are two dangers, two risks are there. So keeping those in mind, let me introduce two groups of two kinds of microscope which worth mentioning. Of course, there are so many other different kinds of microscopes out there, but yeah, I don't have to introduce them to simply to impress you how much I know about those microscopes. What's the point? Okay. So these two and these two group. One is a light microscope and the other one is a fluorescence microscope. And another two that we are going to at least briefly look at it during this course, this lecture, is electron microscope and cryo-EM. Electron microscope is usually abbreviated as EM. So cryo-EM is a type of electron microscope. And actually, I didn't tell you yet, but like this fluorescence microscope is kind of light microscope. So basically what I would like to introduce you at least basic principle and advantage and disadvantage about this. Actually, these two different kinds of microscope. One is light microscope and the other one is EM, electron microscope. So to begin with, we better be able to distinguish fundamentally how they are different. How this light microscope is different from the so-called electron microscope is one thing. And then go further that among these different kind of light microscope, what's so special about the fluorescence microscope? Same thing. Among different electron microscope, what is the big deal of this so-called cryo electron microscope? It's actually my actual real true agenda, hidden agenda that I'd like to share with you during today's lecture. So this is the different parts of light microscope, the standard shape outside the shape of a typical microscope, and these are some of the more fancier, more expensive, advanced microscope in these days. But probably if you were in the freshman general, excuse me, general biology lab, probably you'd struggle to identify each different part and try to memorize, which are all useless. Okay? That's really silly thing to do. Why do you have to pay too much detail, unnecessary detail, and even bother to memorize them? So there are different parts that when you actually have to deal with practically, physically, personally, then you will get to know what are they. So different parts compose even this simple, one of the simple microscope. That's the only thing that you need to know. So the reason why this microscope, light microscope, is called the light microscope is they use the visible light as a visualized illuminating source. Otherwise, you cannot see, because human can only recognize the any image within the visual wavelengths of light. So this is a perfect match. So use a light source that we can recognize as a visible spectrum. And the point is using this lens which refract the light, so you can magnify all these images. That's it. Yeah. So a little detail, if you are kind of interested in, you can take a look at some microscope only use a single lens, but more usually standard light microscope usually use two different lenses to double magnify. But double magnification obviously then invert actually the image upside down because of this light angles. And visible light, what do you mean visible light? The wavelength, specific wavelengths the visible light is within this range. As you go farther to the red side, the energy will decrease. If you go up to the UV blue light spectrum, the energy within the wavelengths of light will be higher. That's kind of basic information. Here, to understand the limitation of a light microscope, we have to understand this conflict between magnifying power and the resolution. So magnification, by making your lens thicker and thicker, theoretically you could achieve much higher magnification, right? However, you have only so much to magnify a certain image without eventually losing this resolution. For example, I just colored these two lines on this ruler with a red pen. But if I keep magnifying this image, eventually this two red line under your magnified image will stick together and then won't be able to actually distinguish which line is which. That's where your resolution limit. So in other words, to simply put, if you keep magnifying under the visible light any object, you will soon reach to some kind of limit that beyond that you cannot really distinguish the object very clearly because of this resolution loss. So that's the basic standard problem of all the light microscope. You can only magnify the image so much. There's a limit. Okay? So that's why generally light microscope, relatively speaking, has a low magnifying power because of this intrinsically poor resolution. To explain a little more detail with some formula, of course I can do that, but that is not necessary. Simply by understanding, okay, light microscope, you cannot really have too much high magnification because of this poor resolution due to mechanical limitation of this light microscope. That's it. That's all you need to know. And another problem is the staining. As I have already explained, you have to introduce this staining in order to actually really clearly visualize any sample specimen. But then the life of this sample specimen will be sacrificed. So, poor thing. So this is one actual real sample example of any specimen examined under the light microscope. The bright field means standard visualization environment. So this is one typical plant cell under which you can recognize this is a really extremely high mag by the light microscope standard, but still you can distinguish nucleus and some other little things which are actually the chloroplast, which is doing the photosynthesis, but other than that, you cannot really see any other too much detail under this. That's it. So you can, although you can play some trick, like so-called phase contrast, then okay, the same image, but if you switch into so-called this phase contrasting mode, then now you can see a little more distinguished structure. But still, are you satisfied? If you were interested in, I'm curious about fine structure of a plant cell, then your curiosity satisfied with this image? Probably not. That's it? Is that it? How can I go further? Yeah. Yes.
Why you talk live is cold?
Because this is a light image, okay, without staining. It's a static plant cell, but let's say if you are trying to see any living cell, moving cell like bacteria or animal cell, or would you be able to easily locate and see them? And another thing is the fine detail. So this is, without staining, this is as much as you could see. Now, let me present this stained version of animal cell. So now you can see some differences like a red stained and some of the white blood cell versus a red blood cell, at least you can distinguish. So medical doctors see immediately, okay, oh, there are a whole bunch of the red blood cells and some peculiar white blood cells are there. And also then on the particular sample, if these white blood cells were found more numerously, then doctors will be ready to diagnose potential cancer, blood cancer, leukemia type. That's how you can use this stained sample of light microscope image. Now, in contrast with this light microscope, the fluorescence microscope is capable of doing this live imaging. Although fluorescence microscope, although it cannot usually give you the overall, all the details, all the structural, everything, just like a light microscope can give you with staining, but at least this fluorescence microscope can specify, because tagging any particular object of your choice with fluorescence marker or dye, whatever you call, then you can simply specifically follow that object in the living cell environment. That's really important, significant breakthrough. So by doing so, you can do this, enable the live imaging over cellular structure and any cellular event. So it became, right after the invention of this technology, fluorescence microscope technology, of course it became a huge trend, a boom. So detailed technological things probably you wouldn't like to receive. But so let me just simply put so if any of these images, the reason why you are able to see some color, green or blue, or even in some cases pink, is because you could, by using some of the interacting molecule, or in some cases, antibody also can be used, because antibody can specifically pinpoint and grab only some specific target object, or in some cases, some chemical which is specifically react to a specific component, for example, carbohydrate only. So you can target each different at a time, of course. Or if you are interested in something like, for example, particular object within the cell, you can use some specific antibody, tagged, fluorescently tagged antibody to visualize only that object, the molecule, in the nucleus. That's why you are now seeing the pink object in the nucleus. And you can also distinguish nucleus in blue from the rest of the cytoplasm and some of the particular protein only present in the cytoplasm in green, for example. So, if you were major, probably you'd want to, and you will, personally, on your own, you will go into more details about this technical thing, because it will really help you to go through, navigate your the major course. But for those who non-majors, I would advise against going into further details of the techniques, because it's useless. You don't really have to. Simply, why don't you stop here and okay. So basically, the principle of this fluorescence microscope is what? You can tag only what you want, okay, with the fluorescence. Fluorescence is the color. The beauty of a fluorescence is it can give you the different color, like green, or in some cases pink, some cases red, some cases orange, yellow, you name it. And then you can only follow that particular protein. Usually that happens to be protein, but not necessarily it always has to be protein. Some other type of macro can also be traced like that. And more importantly, in what? In a living cell. Unlike if you want to color stain like this in the light microscope environment, you would have to kill the cell. But now you can do this in a living cell. In other words, if you are interested in the movement, time-wise movement of a particular protein in a cell, then you can do this by tagging only that particular protein, and while that cell is alive, still you can trace down time-wise and then see some different metabolic activity of that protein too, is the advantage of a fluorescence microscope. So several, those who are like more into the chemistry, basically all those chemicals that can generate fluorescence is usually having this kind of ring-shaped organic molecules. But if you are not, well, never heard of them, then forget it. You won't have to worry about this thing for this course's exam. You know. And there are something called autofluorescence, especially famous in plant cell, because plant cell contains so many chloroplasts in which the molecule functioning as capturing the light, they are naturally fluorescent. So if you examine this same plant cell that you have seen under the light microscope, if you place them under the fluorescence microscope, it will glow automatically, this red light. That kind of thing we call an autofluorescence. And this is actually highlight of the fluorescence microscope. It's called GFP because it stands for green fluorescence protein. And the protein, the gene for making this particular protein GFP only can be found in this guy, jellyfish. That's why jellyfish can glow in the dark. So, of course, people store the gene, this gene, GFP gene, from jellyfish, and then decide to use as one of these fluorescence tag, this protein. The beauty is this protein is a natural protein, so if you somehow combine, we call that a fusion. So now we can fuse, if this was a target protein of your interest, we can make some kind of chimera, artificial chimeric protein, having GFP at the tail of your target protein, that protein you are interested in to pursue. And this stands for the head and tail of a protein, any protein. If you already took your finish your homework of viewing the Bogang makeup lecture, probably you would know already what this NH and COH means, but probably a lot of you haven't done yet. So usually this is a kind of a usual way of designating the end. This is the head, the first, and this is the tail of any given protein. If we align protein in a linear way, although proteins do not lay like linearly like this. They have some very unique specific folding, but let's assume that proteins are there in linear fashion. Then we can usually put this NH amino, we call amino, and there's their head, the first part, and this carboxyl COH, and there's a tail. But anyway, so in other words, you could place you could fuse this GFP protein to the tail end of your target protein, or even you can place your GFP to the head, or head of actually your protein. It doesn't really matter in most of the cases. Most. And if you do so, then now this chimera fusion protein, now if you put this protein inside of the cell that you are going to investigate the behavior of your target protein, then you can follow your target protein. Obviously, naturally, your target protein must have been invisible under the microscope, but now you can see your target protein only thanks to this GFP, green fluorescence protein tagged, tagged. So you can, wherever this green fluorescence light lit up, then that means your target protein is there with them. And also, the thing is, you may wonder, okay, if you generate this type of unusual chimera protein, then the normal, natural function of this target protein would have been disrupted? No, usually no, it's not the case, it turns out that. So usual function, the natural function is preserved. Why? And simply you can follow the protein under the fluorescence microscope. That is really nice thing. So that has been the really tremendous technological breakthrough. So I thought this was like the understanding life science. Maybe I was in the wrong classroom. I thought it was something like a technological understanding technology class. Yeah, but so you can distinguish some of the different locations by employing this trick, which is not really important. And also you can play this type of tricks. You can generate so many different fluorescent mouse. And also you can expand this color repertoire. Originally this was the only color available, but nowadays people wouldn't stay there, so they have generated all different versions of this green fluorescence protein. So now you can, the rainbow color fluorescence proteins you can apply all together at the same time. All kind of different tricks. Yes?
How can we build this GF target protein?
How can we what?
어떻게 GF랑 프로틴을 합칠 수 있어요?
Oh, at the gene level you do. Gene level, you can place this part of the gene, DNA, together with the GFP gene, the DNA together, and then place them, introduce them into the cell. Then that gene is expressed, the gene is converted eventually into the protein, which is a chimeric protein. So now what happened? So these three scientists were the pioneers of developing these techniques, and approximately about 10 years after their first the first report of the success, then all these people were eventually awarded to the Nobel Prize. That means maybe the GFP, green fluorescence protein, was that much important? Yes, it was. It has so much contribution in the advancement of the life science, and in particular this medical science. Okay. So irony is even these two scientists are not here anymore, no longer with us, though. That's one irony is though. So these are one of the selected images that you can see under such fluorescence microscope. This one, you may think, what? Why the broccoli is here? It is a part of your kidney tissue that only in your kidney on a particular type of tissue. It's been labeled with this particular green fluorescence to distinguish it in their distinctive function in mind. And these are actively dividing cells. What do you think? Do you think these cells are undergoing the mitosis or meiosis? What do you think? At least a major, at least a bioscience major should be able to answer this. Here, this golden thing, all these golden stains represent here some of the so-called, how can I forget this? The string that, I can't believe that I forgot the name of this. Maybe later. But anyway, these blue things are chromosomes, and those spindle, yeah, spindle. I'm sorry, thank you. Spindle fibers, the proteins that pulling these chromosomes, the golden things. So these are actually one cell divided into two and then undergo further division over four, which is typical stage of meiosis. In meiosis, cell divide successively, continuously, twice to generate 4 cells. Whereas mitosis, do you even know the difference between mitosis and meiosis? Hopefully? No? Last week we went over mitosis versus meiosis. Okay, then forget it. And these are a particular a nematode, a worm called a nematode, which is really useful for studying the animal development. And these are all different parts of plant cell. So anyway, so many meaningful, beautiful imagery, live imaging can be generated using fluorescence microscope. That's why in modern days, actually, most of these cell biology stuff are done, the investigation, research are done together with this fluorescence microscope. And even the technological advance doesn't stop there, so then more recently, even using this fluorescence microscope, resolution has reached to this stage so that even these little dots are even the scale of individual ribosome. That means individual ribosomes can only be seen even really high scale electron microscope before, but now even you can by using fluorescence microscope is basically a type of a light microscope. So light microscope limitation is what? Resolution. So you cannot magnify too much. But who says so? Even you can now increase this magnification to this scale with this detailed resolution in these. It's really shocking these days. The rate of this technology advancing, as we cannot really catch up. And all these are actually made, all these contributions were basically made not by pure biologists, but guess who? Basically those who, the physicist or the chemist. They are the one who gave us this tremendous help in the advancement of the quest for this light mystery. And hold on, please. 여보세요? 저 조금 더 전화 드리면 안 될까요? 제가 수업 중이라서. 예. Now, this is electron microscope. Electron microscope is, as opposed to this visible light, use an electron beam as a source of this magnification, illumination. So by doing so, there is some secret that I don't really have to, it's not a secret, but some detail, technical detail, I don't really have to go over here. But because of that, you can tremendously increase this magnification without losing, without sacrificing this resolution. So in other words, simply put, electron microscope can give such a huge high magnification with a high resolution. This is very good, of course. So, however, that's an upside. Then what's downside? It's so costly. Not a single man usually can easily afford having this electron microscope. Anybody is actually in this department, any professor having their own electron microscope in their lab? No, nobody. That's a huge, expensive, really cumbersome instrument. And also, it takes a lot of training to actually correctly, soundly prepare. Usually some trained technician can only handle this sample prep because it deals with some of the really detailed, time-consuming training is required for this. And actually more importantly, this. During the sample prep, you have to treat this sample very specifically to increase the contrast. So a lot of artifacts. So you may actually, at one side, at a site, oh, you're so impressed with, oh, this is so much beautiful details of inside of a cell, but actually most of them may not actually something that naturally exists. It's always that's a really serious drawback of the EM image. So EM looks like this. And so this too. You have seen this light microscope image before. And this is relatively very low magnification of electron microscope. There are two different types of electron microscope. One is called transmission and the other one is scanning. You don't really need to know the actual technical details of the differences between these two. But anyway, so you can now see some of the details. And like I said, this is only very low end of the magnification. So you can really expand this so that you can see really fine details of inside the structure. And also in this occasion, you can see the surface, 3D structure of any object. So that's the beauty of electron microscope. So here, really real details. And the other one is, although previously, another drawback of electron microscope is you can only see the black and white image under the electron microscope. But nowadays, who says so? Computer can color everything. So once you generate this black and white original image, then computer will colorize this. And this is a computer-generated colorized version of the EM, which is here the nucleus, and chromosome is there, and here the ER, endoplasmic reticulum, that we are going to see later on. What are they? So one of the organelles in the eukaryotic cells are shown in really very much detail. That's the beauty. And speaking of scanning electron microscope, you can see, you see what this is about? Bacteriophage, that virus that infects the bacteria. So this must be a bacterial cell surface, and one virus is now attached on to the surface of this bacterial cell, and then trying to now go in. The image was captured. And this is a mouse part of a fly. And this is pollens. And this is your inside of your blood. They are all real. Some of them are color coded, but they are all real image. Red blood cell and white blood cells. So you can see the power of the electron microscope. But still, those disadvantages that I mentioned, the drawback of electron microscope is too much. Although it can provide you with this detailed information, but usually you don't really want to go to the step of having to go through with the electron microscope unless you really have to, because that's too much pain in your behind. That much disadvantage is there. Now, some different version of the electron microscope can even provide this much details of even one DNA strand and some surface of this graphite image can be seen under the electron microscope. So big deal, fine. Now, the real purpose of today's topic is this. Cryo-EM is what kind of EM? But cryo means freezing. Freaking freezing low temperature is a cryo. So that means this EM is being operated under extremely low temperature is what it means. Cryo-EM. And so this is a specialized electron microscope, of course. And this must be operated under very low temperature. And the purpose of this cryo-EM is not visualizing the details of cell structure. No, not anymore. This cryo-EM's objective is elsewhere. Structure. Structure, whenever we mention structure, that usually automatically at this point in these days, we refer to the protein structure. Protein structure is really extremely important because of the functional, direct relationship with their function. So everybody wants to know the protein structure. Guess what? This year's Nobel Prize winner was what? Not the literature, but the other science area. AlphaFold 2. Have you ever heard of this AlphaFold 2? This is a protein structure predicting artificial intelligence model. This AI was awarded the Nobel Prize this year.
Wow.
Wow, big deal.
Why 알파포드 AI가 왜 높은 화학
I didn't even bother to know whatever, big guy, who cares? But yes, they are affiliated with the Google, the AlphaFold. So anyway, AlphaFold is the specifically developed the artificial intelligence for this particular purpose of predicting and presenting some predictive structure model of a protein. And with regards to, and guess why this one was regarded so significantly so that even people had to give a Nobel Prize, prestigious Nobel Prize to them. And this one, cryo-EM was actually the previous year's Nobel Prize winner. Those who developed this cryo-EM. So these days, a structure revealing, elucidating, so to speak, the protein structure is a really big thing in many different areas. In particular, in medicine, trying to develop new drugs and trying to develop new antibodies. It all takes that having correct information about particular target protein's structure. Why? Structure dictates function. Don't forget, in biology, in life, structure dictates function. Shape matters. It's no big, deep secret, but simple. So anyway, how this is like the principle, who cares? Oh, briefly speaking, the principle of this rationale of cryo-EM is in that under extreme low temperature, you break down proteins into pieces, fragments. So you have to collect multiples of so many, the same kind of protein prepared in pure form, and then freeze them under extremely low temperature, and then each individual protein will be broken into smaller, much smaller pieces, and they will be scattered into all of the place over there in random way. So think about it. If I was broken down into small pieces, just like a piece of glasses, because of this too low temperature, then this part, all these different parts of me will be laid upon on this flat surface in different random fashions. So if me in copies, multiple copies, was presented in the multiple copies of fragments, then computer scans this shape and then recombine and reconstruct the overall, the entire native shape is the AI aided computer's job. So that's how this cryo-EM can provide some very accurate structural information of any given macromolecules. It doesn't really have to be proteins, but usually people are more interested in the structure of the protein, and that's why. So, it was even several years ago, 2017, these three scientists who collaborated in developing this cryo-EM technology was finally recognized and awarded of Nobel Prize. And this is showing some progress of structural fine details, but more importantly, anybody recognize this famous figure structure? What is this? Coronavirus. Surface of a coronavirus. And remember that somehow people were able to so quickly develop a vaccine against this coronavirus. Usually, if we took the conventional way of a vaccine development, it would have taken minimum 2 or 3 years, but was developed generally within even less than a year, several months. How? Using this cryo-EM to get the structural information of a particular area of this here. This was target on the viral surface as target for the vaccine, okay, antibody. So specifically obtaining this fine detailed structural information like this, then scientists were able to screen correct structurally fit antibodies against it, and that's how they were able to generate this thing. So it has actually a tremendous contribution to this. Although still, in the meantime, the human in general suffered greatly from this coronavirus pandemics, but on the other side, the scientists, the struggling effort has been there to compensate such problem. Okay, so that was about microscope. So, have you gotten any take-home message about microscope then? And also you were able to understand my intention why that guy introduced such a boring topic of microscope technology in the beginning of the cell lecture. So now at least, probably, hopefully, you would be able to understand better about why in these days, whenever you just turn the page regarding any magazine or scientifically related the information or the scientific journal, then why you have so many just fluorescently looking images and also this kind of structural things and cryo-EM, blah, 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 fluorescence imaging, blah, blah, blah. These two, fluorescence microscope and cryo-EM are the two actually wings leading this advancement of this, at least cellular aspect of modern biology. So at least if you understand, okay, that's how the fluorescence microscope is basically this. This is how it is being operated. On the other hand, cryo-EM is what? Doing this. And what's the purpose of this cryo-EM? The advantage and disadvantage and actual purpose of each of these two, if you can correctly understand and distinguish, that will be, I will be happy about. Okay, so now we can go on to prokaryotic cells. Okay, about 15 minutes left. So let me just first, it's been a while, so let me take care of all, only few of, always it's such a dilemma that at the end of every lecture, I receive so many valuable, interesting questions, but only I have so much time to go over this. So those questions that were selected to be presented over here is not necessarily is because those questions were like worse and those that are left were not really was so much. No, that is not the case, but only because of this randomized time limit, I only had to choose only a few of them. So I'm keep thinking how am I going to be able to make up for those unselected questions? Still keep thinking, but anyway, for now, just focus on this. What do you think about this? Do you have any? Your own thought? Some environmental effect, and there are some effort to adapt to this changing environment. So how usually this adaptation will go, or is there any general trend for any those adaptive response on the changing environment is basically the theme of this question. Like, this is actually the answer. The correct answer is I don't know. Yeah. But it depends. For example, this question is a question asks that if this climate change, the changing the temperature to colder environment, so that having more furs, more hairs became suddenly a little bit more selectively advantageous. But then those having, no matter how much you just are equipped, so eventually equipped with this hair, but still eventually you are not able to overcome this like freezing temperature. So then does that mean if you compare these two people, that those who were eliminated in the early stage of this colder temperature versus those who stays till the last and then only succumb to the last blow of this colder temperature, if you compare those who were at least having a little more fur, logically, yeah, it could have been. But depending on this period, what really more important about this question is how quick was this changing environment was. If the changing colder temperature was gradually went down, then probably over several generations you might be able to see some trend. But if those changing in cold temperature was so quick, then the people didn't have enough time to adjust in terms of transmitting their different traits into the next generation. So that's probably what makes it different. Long question. Cute drawing, I like it. The thing is only I'd like to focus this. Yes, the human Y chromosome, probably one of the only utility of human Y chromosome is maybe this gene. It's called SRY. The name of the gene is SRY, whatever. But this gene is responsible for determining the maleness. Okay. And it is only present on the Y chromosome. So if you actually intentionally, artificially transplant this SRY gene region into X chromosome, those individuals having only two X chromosome, which is biologically female, but biologically speaking, you can change this female into male. Although it was confirmed in the experiment of a mouse. Obviously, you cannot do this thing in human, of course, but at least when you try this thing in mouse, it changed the sex of this mouse. It should have been having two X chromosomes, it should have been female mouse, but by obtaining one extra SRE gene from Y chromosome transplanted on Y chromosome, this mouse became male. So that actually demonstrated that this SRY gene is the one who is responsible for this determining the maleness. Okay? The radioactive isotope is why this is radioactive is because the isotope it's been explained, I believe, in the makeup lectures, but I will reiterate this, that isotope is the one having heavier, more number of the neutrons in the nucleus. So it's a heavier version of the so-called more general, more common form of element. But depending on how many more neutrons in the nucleus this element has, the higher energy, the more you have this, become heavier and more unstable because of too much energy in the nucleus. Then it becomes radioactive because too much energy and try to stabilize, try to come back to the general standard level, then it will emit the energy in the form of radioactive thing. That's what we call the decay, of radioactive decay. How? They were all long time ago, they were all gone, extinct, and how were you able to collect all those DNA from their fossil, the bone fossil, and then reconstitute to complete the whole genome sequence? What's the questions? Yeah, it was amazing job. Probably you must have heard this technology because of this corona test, PCR technique, was the actually play the pivotal role. Because once you have only very trace amount of small pieces of DNA, collect this, then you can amplify the huge amount using this technology. So you can collect, of course, you cannot recover entire intact DNA from one individual fossil of Neanderthal, but you collect some of them from this fossil, and you are able to collect some other from the other. And then you collect all of them, although they are all very tiny amount, But if you place them under this PCR technology, they can be amplified tremendously. And then that's how we were able to reconstitute. Mitochondrial DNA is inherited from mothers only. What's the general role of this mitochondrial DNA, right? And what kind of effects would this mitochondrial DNA have on our nuclear DNA? It's a very good question. Mitochondrial DNA is originally from individually living microorganism. Okay, so this mitochondrial DNA is the genes originally present in mitochondria. Something to do with their function of their metabolism, which is actually the ATP. Most of them are ATP metabolism related gene. And also some other genes are for their own protein functions. And actually, surprisingly, a lot of them actually, majority of those originally, those ATP metabolism related genes originally present in the mitochondria have already moved into the nucleus during the course of this the living together. But still, some of the genes that still remain there and then functioning inside. And those who have left, just like the human migration from Africa, is very similar. And those who now present in the nuclear genome, those protein products are coming back to the mitochondria, inside of mitochondria, to have a function together, even though they have moved their presence into the nuclear gene. That's their relationship. Oh, this is called grasp reflex. The babies, if you just reach out your finger, babies just instinctively just will grab it. And these grasp reflex are usually found on the mammals, especially those like fur-having mammals, like the chimpanzees and other monkeys, and those animals that carry their babies on their back or on their chest. Usually, those babies have this type of reflex instinct. And probably you could easily imagine what kind of a benefit would this kind of a reflex would have provided by being able to grab babies, and then the mother can lift a little bit of a burden to. Otherwise, mother always had to carry their babies with their arms or their mouths are occupied. But by doing so, they can still cling to their body part. So both of them can survive their chances. This YXX individual, it's called Klinefelter syndrome, having 2X chromosome and 1 Y chromosome. What do you think? The actual biological sex of this individual, what do you think? Would this individual be male or female? Why?
Y chromosome.
Yeah, because of the Y chromosome. Be biologically male, but not entirely 100%, so to speak, standard male, because of the presence of these two X chromosomes. I will give this question on your midterm exam. Guess why? How is it possibly generated, this YXX? So with this, I have another student question that I promised that I'm going to put that on your midterm exam. There were two the midterm exam related your questions by now generated, right? Okay, so think about this. And you should be able to draw how and what mechanism this YXX can be possibly generated. I believe I gave you enough time to research this, to correctly answer this. So it shouldn't be any of a problem. Okay, so with that, I should stop today's lecture then, I guess.